In this video, we're going to look at the structure of the 20 common amino acids. We're going to look at how amino acids are combined through a peptide bond to make polypeptides. And we're going to look at how some amino acids can be charged and their pK or pKa values. Now, all amino acids have the same standard shape. They have an amino group connected to a carbon, which we call the carbon alpha, and they have a carboxylic acid group. And they also have an R group, where R is 20 different things, so R is different for each 20 amino acids. And attached to this carbon alpha is also a hydrogen. Right? And we say 20 standard amino acids because in general 20 amino acids make up every single protein on Earth. There are other amino acids, a lot more amino acids, but we're going to focus on the 20 standard ones in this video. Now, inside the cell, which has a pH of roughly 7.2, amino acids are what are called Zwitter ions. Zwei is the German word for two, and ion. So this NH2 group under cellular conditions is NH3 and is positively charged. This COOH group is COO minus under cellular conditions and is negatively charged. So what you need to know for me when it comes to the 20 amino acids, you need to know the full name of all the amino acids. You need to know the 3-letter code of the amino acids. You need to know the 1-letter code of the amino acids. You need to be able to draw the structure of the 20 amino acids. If the amino acid has a pKa for the side chain, you need to know that number. And you need to know that in general, all N-term is where the nitrogen is, pKa is roughly 9.4, and all C-term, that's where COOH is roughly 2.2. Now we're going to start looking at the structure of all of our amino acids. And first we start with the two amino acids that are negatively charged under cellular conditions. That's aspartic acid and glutamic acid. All right. So first, let's just look at the naming scheme for these amino acids. I mean carbon naming scheme. So the carbon that is connected to the N and C on our what's called the backbone, we always call that the alpha carbon. And then the carbon groups attached to it as we move up get a letter in the Greek alphabet. So we go to alpha, beta, gamma, and then delta, and epsilon if we had another carbon, so on and so forth. So aspartic acid, the pKa or pKr, the side chain, is 3.7, and for glutamic acid is 4.3. Like I said, you need to know those. The specific numbers down here, again, you don't need to know the specific numbers for the backbone for each amino acid, just the average numbers I gave in the previous slides. And if we look at here, what this is, it's called a ball and stick representation of amino acids, and it's telling us the charge distribution. So red means negatively charged, blue means positively charged, and green or white means neutral, right? So here's our backbone, COO minus, carbon alpha, NH3, and here's CH2, and here's our COO minus of our side chain. That's aspartate. And I'm going to go over some tips and tricks that biochemist students use to remember what are called the 3-letter codes and 1-letter codes of amino acids. So in general, I can use 3 different names to call this amino acid. I could call it aspartic acid or aspartate. I could call this amino acid asp, or I could just call it D. And talking to biochemists, if I use any of those names, they know what I'm talking about. So that's why it's important for you to know those names. Okay, so the tricks I've learned for the negative amino acids is aspartic acid. So the way I pronounce aspartic is you really stress the D in aspartic acid. So that's why it gets the one letter code D. And to draw it, if you look at the side chain, you can think of D as di for 2, right? So it has 2 carbons. While glutamic acid, the one letter code for glutamic acid is E. That's because the 3-letter code is glue, right? Glue can be spelled Glue. So glue for glutamic acid. And to remember how the side chain of glutamic acid looks like, E is 1 letter after D in the alphabet. So glutamic acid has 1 more carbon in its side chain than aspartic acid. So those are our 2 negative side chains. We have 3 positive side chains: lysine, arginine, and histidine. Now, again, remember the pKa's for these 3. And I say kind of here. Histidine sometimes is charged inside of a cell, and it's sometimes not. I think 30% of the time is positively charged, 70% of the time it's not, and that's due to its pKa. But we'll talk about that later. Right now, let's consider it positively charged. So I don't have a special trick for lysine being called K as its first letter. I just, for some reason, always remembered special K being lysine, you know, the cereal special K. So lysine is special K. Arginine is easy for a 1 letter because of the way you pronounce arginine. You really stress the R, and the lame joke that goes with that is, what is a pirate's favorite amino acid? Well, that would be arginine. And histidine, well, histidine is the only amino acid that starts with an H. So I guess the 1 letter code of H. And histidine is a ring. Arginine has 3 nitrogens in the side chain. It's the only amino acid that has that. And lysine has an NH3 group at the top. Now let's go to the side chains that have a polarity but are not charged, are polar non-charged side chains. So we start with serine and cysteine. Notice serine and cysteine look very much the same, except serine, letter code S, has a hydroxyl, while cysteine, letter code C, has a thiol. That's the only difference. And cysteine, we'll look at this later in the course, but cysteine has this special property of when you take 2 cysteines and you point their side chains at each other, you form what's called a disulfide bond. So 2 cysteine side chains will react to form a covalent bond called a disulfide. Alright, now the next polar non-charged are asparagine and glutamine. So asparagine gets the letter code N because the 3-letter code is ASN, so asparagine. And glutamine gets the letter code Q because it's just the cutest little amino acid you ever did see. Yes, it is. It's cutie, glutamine. Now, I'm making a fool of myself here because I want you to remember these. So if you remember, oh, Dr. Andrews says glutamine, it's dumb, but it makes me remember Q, then my job is done. Now, for what asparagine and glutamine looks like, we go back to our negative amino acids. So you remember aspartic acid and glutamic acid have C double bond O's at the top. Well, all you need to do with asparagine is take one of the oxygens from aspartic acid and put NH2. From glutamine, take glutamic acid and put an NH2. That's why it's called glutamine. It's glutamic acid but with an amine. Asparagine, it's the same idea. Now threonine, take serine, add another carbon to it, and you get threonine. So add a carbon to serine here to get threonine. The way to remember that is, again, I go with the alphabet. T comes after S in the alphabet, so threonine has an extra carbon compared to serine. Now let's go to our aromatic side chains. We have 3 aromatic amino acids. We have phenylalanine, tyrosine, and tryptophan. So phenylalanine is easy amino acid to remember what it looks like, because if we remove this phenol group, we would just have alanine. So alanine is just a CH3 right here. We add a phenyl group to alanine, we get phenylalanine. And F, our one-letter code of F, is because how we pronounce phenylalanine. You really stress the F in phenylalanine. That's why it gets the one-letter code F. Now take phenylalanine, put a hydroxyl on phenylalanine, and you get tyrosine. Now the way to remember tyrosine is a Y, is the way you pronounce tyrosine, and you overstress the Y again. And our double ring structure is called tryptophan, and it gets a W because it is double ring, tryptophan. And lastly, we have our amino acids that are hydrophobic or nonpolar side chain. The simplest amino acid there is, which only has a hydrogen as its R group, is called glycine. And then if instead of a hydrogen you have a methyl, CH3, like we talked about in the previous slide, that's called alanine. Proline is a very interesting amino acid because proline is the only side chain that wraps around on this NH3 group. So the side chain actually binds to the backbone to make NH2, and you get proline. Valine has 3 carbons that form a V in the backbone because it's valine for V. While leucine, L, is a little bit longer than valine, and since it's a little bit longer, it gets 1 extra carbon for leucine. And isoleucine is if you take leucine and switch the position of the CH3 group and you move it down 1 to make isoleucine. And finally, methionine. Methionine is the only complex in it other than cysteine to have a sulfur. So you have 4 side chain groups: carbon, carbon, sulfur, carbon. CCSC. That's for methionine. Now, in general, most amino acids aren't found alone inside your body. They are found inside proteins. And the way you combined amino acids is a reaction we've seen before. It's a condensation reaction. So you take the C terminus of one amino acid and you combine it with the N terminus of another amino acid. And basically the oxygen and 2 hydrogens are coming off, so you're condensing water and you form a new covalent bond. This bond is called a peptide bond. And you can make dipeptides if it's 2 amino acids, tripeptides if it's 3 amino acids. Oligo and polypeptides are just longer and longer amino acids. But for these peptides, the residues are amino acids. And when you're working with proteins or peptides, you always start at the N terminus and you move to the C terminus. And that's how you read amino acids. For example, we have the sequence of alanine, tyrosine, aspartic acid, and glycine. So I would call this peptide AYDG based on its 1-letter codes. And proteins, which we'll talk about here in a little bit, proteins are just molecules that have 1 or more peptide chains in it. Now, as we saw in the side chains of amino acids, we have some amino acids that are negatively and positively charged and some that are polar but non-charged. And so every amino acid contains ionizable groups, either 2 or 3. Now, let's look at what this means for our amino acids. So let's imagine that our pH is 1. We're in a very acidic condition. If you're very acidic, that means you have a lot of H3O plus around. Therefore, everything is protonated, and your backbone or your C double bond O groups would actually be COOH in neutral, and your NH3 groups will be positively charged. While if you're at high pHs, your pH will be, let's say, 13. That means you are in very basic conditions, which means everything will be unprotonated. So this COOH becomes COO minus. This NH3 plus loses a hydrogen and becomes NH2. Now remember, a hydrogen has a +1 charge, so when you go from NH3 plus, you lose a hydrogen, you form NH2. You go to COOH, you lose a hydrogen, and you become COO minus. So for amino acids, if we know the pH, we can predict the charge of the amino acid. It's a little more difficult if you're at neutral pH. But in general, and we'll do some of this in class because it's kind of a hard concept to get just talking about it. In general, if you look at the pKa or pKa of your ionizable group, if your pH is bigger than that number, your ionizable group is unprotonated. So for example, if we're at a pH of 7 and the pKa of our C terminus is 2.2, it is unprotonated and negatively charged. While if your pH is lower than your pKa, your group is protonated, right? So the pKa of our N terminus is roughly 12. At neutral pH, we're below that, so we are protonated. And again, we'll do some practice of that in class. Now, let's talk about the idea of isoelectric point. Isoelectric point, or PI, is when a molecule is electrically neutral. And we have a simple way to determine this for amino acids. The value of your PI equals 1/2 of pKi plus pKg. So what's pKi and pKg? So for neutral amino acids like glycine here, pKi is simply the pKa of your N term. pKg is the pKa of your C term. So here we have glycine, right? So pKj here is 2.34, pKi is 9.60. Plug them into this equation, and we find that glycine is neutral at a pH of 5.97. So that's what PI means. It's what pH you're neutral at. For charged amino acids, our definitions are different. pKi is your C terminus, pKj is your side chain, and for positive amino acids, pKi is your side chain and pKj is your N term. Now, unfortunately, if we're working with peptides, this equation doesn't predict it like it does for single amino acids. And the reason for that is in a peptide, your side chains are going to be interacting with other charged or polar molecules, and because of that, your pKa's shift, and that really has to do with a lot of physical chemistry, which this is biochemistry and not physical chemistry, so we're not going to get into that. But we're just going to say experimentally it's difficult to calculate PI for peptides. And that's it for this video on amino acids. I will see you in the next video.